Okay, good morning everyone. So today we'll be discussing about Phomopsis blight of brinjal or eggplants. So you might have heard about Phomopsis blight because it is one of the most important diseases of maize, of brinjal, and it can be found uh, in severe cases in Sirat, Sarlahi. Those are the major brinjal growing areas. So, Phomopsis vexans is the causal organism. Its telomorphic state is diporthe vexans. So, this Phomopsis, we'll discuss about alpha and beta condia of Phomopsis. So Phomopsis, when the infections it takes place in nursery stage, it can cause damping off. That means it causes infection on the collar region, and there will be development of some brown colored streak on the collar region that will enlarge in size, and everything is girdled and plant will topple over. And on leaves symptoms, they are very confusing with Cercospora leaf spot of brinjal. So there are few important things that can be considered for the diagnosis of Phomopsis blight based on observation of leaves, infected leaves. So generally, as you can see in this photograph, the lesions, they are generally more or less circular to oval. And in the initial stage, the entire lesion may be white or it may be grayish, but soon the margins of the lesions it becomes dark colored, or it becomes reddish brown, or sometimes totally brown, and due to which the circular spots, they have gray center. That means they have light color center and dark margin. And with the help of microscope, on the gray areas, one can visualize pycnidia, and you must know what is pycnidia. To visualize pycnidia, they are just flask shaped black colored asexual fruiting body. So these spots, they will enlarge in size, they will coalesce, and they cause premature defoliation of the leaf. So characteristic symptoms, they are observed on fruits. So if infections, they are very severe, on a single infected eggplant, lot of mummified fruits may be visualized. But this is advancing phase of symptoms on the fruit of Phomopsis blight. So generally, they infest as some kind of discolored sunken spot, which are generally pale brown, and with enlargement of these pale brown sunken spots, number of dark colored pycnidia, black fruiting mass, they may be formed. So here you can see some black dot like things which are arranged in concentric rings. This is in case of stem, and when they coalesce, they form irregular patches. And in this photograph, you can see again some dot like structures, some minute dot like structures. You can also see some zonation. So there are so many variable symptoms, and it is always affected by environmental conditions and virulence of the the isolate of Phomopsis. So this is when there is infection on the calyx, then it causes entire rotting of the fruit, and the brinjal fruit, it remains mummified, and it remains attached to the brinjal plant for a long period of time. So this is one of the very important disease of brinjal. And I was talking about the conidia. So this pathogen, it produces two kinds of conidia. One is known as alpha conidia, and another is known as beta conidia. So beta conidia, they are filiform. They are just like needle. They are slightly curved. They are also septate, but you won't be able to see the septa in this photograph. And beta spores or beta conidia, they are always formed at lower temperature conditions, that is at 10 to 16 degrees Celsius. While alpha conidia, they are aseptate. They are typically one-celled. They are subcylindrical, and they are generally produced at higher temperature during summer months. So both kinds of conidia, it may be retrieved from the infected plant parts. So they have two vacuoles. So in this photograph, you will be able to see it much clearly. So they are oil globules. They are bigutulate. They are subcylindrical. These are beta conidia. They are filiform. They are septate. While alpha conidia, they are subcylindrical. They are bigutulate. They are one-celled, and they are very small. So this is under this is under higher magnification, and when you observe this under lower magnification, they just seem some elongated dot-like thing under microscope. So so this pathogen, it is both seed-borne as well as it can survive saprophytically on infected plant debris as dormant mycelium. And it is this disease is very severe when temperature conditions, they are very wet, and it's around 26 degrees Celsius, 25 to 20 degrees Celsius. And this also gets established in the fruits as a latent infection. So again, when the brinjal, they are taken out of the cold storage, and on the after storage conditions, symptoms may be seen due to latent infections. So these are some mummified fruits. You can see some sunken pale areas, that is again accompanied by some zonation-like effect with profuse formation of pycnidia-like thing. Or these are black fruiting bodies which are known as pycnidia. So about management, Pusa Vairav, it is resistant variety, and Florida market, it's not available in our case. So hot water treatment, it is not advisable to the farmers, and it can be only it can be carried out by professionals. So generally, seed treatment is advisable with carbendazim at the rate of 2.5 gram. And when there is high, or when there is severe cases of this disease, crop rotation with non-host crop is generally advised, because it can persist in soil for a couple of years. So seed treatment with carbendazim, it can be done at 2.5 to 3 grams. Again, root seedling dip. So this is root seedling dip. So root seedling dip is also found very effective, and it is done at the rate of 0.05% solution. That means 0.5 gram of carbendazim per ml, and it should not be carried out for more than 30 minutes. Generally, we advise to carry root seedling treatment for 15 to 20 minutes. And again, on initiation of the disease, one can go with foliar spray of carbendazim or even thiophanate methyl. So this is about the management of one of the important diseases of brinjal. So next is Cercospora leaf spot of brinjal. So the symptoms on the leaf, they are very similar to Phomopsis leaf blight or leaf spot that are produced by Phomopsis vexans. So in case of Cercospora leaf spot, there are 3 species reported to cause this disease: Cercospora melangina, Cercospora eugenila, and Cercospora solanigena. So all of the species, they have some, they produce some different kinds of symptoms. Right? So in all of the cases, some kind of spot is produced, but the color, it varies as per the species involved in the disease development. So about Cercospora melongena, these are symptoms of Cercospora melongena. These are most typically, or they are abundantly formed on lower leaves, and on the upper leaves, they are, if we compare the infection caused by these 3 species, the spots, they are very large. Relatively, they are very large. They are generally circular. With disease progression, they may become irregular. And just as in case of Phomopsis leaf blight, they also have light colored center or grayish center. It may be grayish brown, and they generally range from 4 to 10 So in case of Sarcospora igenuila, so the initial symptoms, they appear as some kind of grayish white spots. They are very small in size, lot of leaf specks or uh, leaf spot appear, which are generally circular, but they may be also angular. And in this case, short hole effect is very pronounced as compared to the infection due to other species of Cercospora. In case of Cercospora igenuila, short holes effect is very pronounced. They cause premature defoliation of the leaves. In case of Cercospora solanigena, so this is not reported from our context. It's reported from some other context. So this is not important for you. So you must be able to diagnose Cercospora leaf spots. In case of Cercospora melongena, these are typical symptoms of Cercospora melongena. You can see light colored center. They are more or less grayish brown. They have dark colored margin that is typically brown, and when they enlarge, they form large patches. And in case of infections due to Cercospora igenuila, short hole effects are very pronounced. They produce some kind of peppery effect, peppery pale yellow effect on the infected leaves. So they are also seed borne as well as. So this is infection of Cercospora igenuila. So while here it is igenuila, so lot of kinds of both species they may coexist. So this is about the disease cycle. Generally, it's again favored by 25 to 26 degrees Celsius. So Panch Samrat, it is one of the resistant varieties to this disease, and management, it is very common. One can go with carbendazim, thiophanate methyl, or zineb. Similarly, again, alternaria leaf spot of brinjal, it is seen in the fields. It is caused by two species of alternaria, Alternaria melongena and Alternaria solani. Alternaria solani also causes early leaf spot or early leaf blight of tomato and potato, which we will be discussing in some other classes. So in case of Alternaria infection, as you know, the symptoms, they are very similar in all of the crop species. They produce some kind of dark brown spots, and those spots, they gradually develop some kind of zonation or target-like effect. And that is one of the very common symptoms. So you can see here some kind of zonations, concentric ring-like pattern. So those can be clearly visualized when one sees the spot against the sunlight. So again, the management, it is not so important disease in case of brinjal. Little like leaf of brinjal, it is often seen in the fields, generally, particularly in volunteer brinjal plants. It is also seen little leaf of because this phytoplasmid can easily survive on datura metal. So the datura, these kinds of symptoms are seen. So in case of little leaf of brinjal, so there is excessive reduction in the leaf size. There is reduction in the internode region, and there is bunching like effect or rosetting effect to give the entire plant a bushy appearance. And this is systemic in this case. If actually these are transmitted by plant hoppers. So Impoesca devestans र Isimonas phycetis भन्ने दुईटा चाहिँ leaf hopper को स्पिसिसहरु छन्. They are also graft transmissible. They can survive in a lot of weed hosts. as I was talking about Hishimoto's fasciitis and Impois catavistens, so they transmit this phytoplasma. You know what is phytoplasma, and it's a soft-bodied bacteria. It's quite different from bacteria. They cannot be cultured. So, about management, in some cases, this is seen in severe cases. जस्तो अहिले चाहिँ the rice को चाहिँ maturity stage र brinjalको चाहिँ अहिले vegetative stage they coincide. So plant hoppers, they may easily migrate from rice fields to brinjal fields or from weed hosts. So generally we recommend dimethoate, which is available as raw roser at the rate of 0.1%, which is highly systemic. This bag you can go with entomology-ma tapaile पढ्नु होला lambda cyhalothrin. So lambda cyhalothrin is also one of the recommendations for management of plant hoppers. Just the thiomethazam भन्ने पढ्नु होला, that is recommended at the rate of 1 gram per liter because they are very high systemic, thiomethazam and lambda cyhalothrin. So for this we also recommend in areas with this problem, we recommend seedling dip in tetracycline, and we recommend to dip the entire seedling in the tetracycline solution for 10 to 15 minutes. And so that is just at the rate of 0.05%. That means 0.5 gram per ml. That makes it 500 ppm. So I won't be discussing about sclerotonia rot of eggplants because we have discussed this time and again in case of beans, in case of I guess recently we discussed about this in case of pea as well. So everything is similar. So sclerotonia, it has a very wide host range. So they generally infect the collar lesion, which is close to the soil lesion. There is some kind of water soaking, some dark streaks are formed, and soon fluffy mycelial growth is seen, which is accompanied by formation of dark colored sclerotia, and they proceed upward. They can cause infection of the leaves and they can cause blighting. About management, about disease cycle, everything is same. So I won't be discussing about this time and again. Damping off of brinjal. So generally damping off, they are caused by numerous soil-borne pathogens. So sometimes they may be caused by Fusarium solani or rhizoctonia also causes damping off. So even your kharep or your phomopsis can cause damping off, but generally, typically damping off, they are caused by Pythium species, particularly Pythium ephendermatum and Pythium ultimum. So Pythium species and Rhizoctonia species, they are more common in wet soil conditions, while Fusarium, they are abundant in moderately wet soil conditions, because under wet soil conditions, Fusarium, the population gradually declines. So पहिला पनि पढिसक्नु भएको छ यो damping off को बारेमा, about pre-emergence damping off, about post-emergence damping off. There is infection on the collar region, generally hair. So or in case of Fusarium, it may extend slightly up to here. So because of the vascular discoloration, they may initially the plant may show some kind of wilting in case of Fusarium solani. But in case of infections due to Pythium, so infected tissues, they are generally collar region. Initially, there's some kind of water soaking. So those infections on the collar region, it also proceeds to the root, and gradually the stem it becomes constricted at the base and it collapses or it topple over. Uh, in case of fusarium, in early morning, the nursery plants they may seem to recover, but again on during day they may wilt. So those are very general symptoms. So generally Pythium they are favored by acidic conditions, pH of 6.0. They are favored by wet and heavy soils. So general temperature of this season, this the rainy season, it is very conducive. Or winter in just the Brinjal case, Brinjal is already in the field. It's transplanted just a month back in normal condition. So oil case Pythium they may cause damping of if there's high inoculum level in the soil. Post-emergence damping of they can be witnessed in higher temperature conditions as well, 30 to 40 degrees Celsius, but they are not so common. Pre-emergence damping of they are more common. So about soil-borne management, I have repeatedly told you that you must go with biocontrol agents, and I have already enlightened you about how to use those biocontrol agents, because the is not important. How you apply, what's the method of application, that is very important regarding the efficacy of biocontrol agents. Just like urea, you know about urea. So you urea. So there's oxidized zone and reduced zone in soil. So when soil it reacts with oxygen or air, so that becomes volatile, that gets converted into nitric analogs or derivatives of nitrogen. So generally किसानले चाहिँ युरिया माथि छर्या हुन्छ हैन. So that is a very wrong practice. र युरिया छर्दाखेरि soil should be wet भनेर पनि पढ्नु होला तपाईले हैन. So युरिया मात्र छरेर चाहिँ production cannot be increased. We must be very concerned about the application method. So it must get converted into ammonium form for absorption हैन. So अब त्यो सुक्खा खेत छ, पानी नै छैन, पानी ला छैन, त्यतिकै युरिया छर्या छ, that is useless. अथवा चाहिँ माथि छर्या छ, that is useless. So त्यो कुराहरु तपाईलाई एग्रोनोमीका सरहरुले एकदम धेरै राम्ररी पढाउनु भयो होला, धेरै विज्ञ सरहरु हुनुहुन्छ एग्रोनोमीमा. So I won't be discussing about those things. So generally Pythium they can be managed with metalaxyl, metalaxyl formulations. So about I missed one of the disease in case of crucifers. We will also be discussing about this disease in solanaceous crops too. So you know about alternaria infections, so generally in case of solanaceous and crucifers, so solanaceous ko pani ali bhaninchu. Generally in crucifers, alternaria brassicae and brassicicola they are involved. So there are different species. Alternaria raffani, it generally infects radish. So in case of infections, this is infection of Alternaria brassicicola. So the spots which are found by Alternaria brassicicola, they are more or less dark brown, and with advancement of disease, they may get black colored, right? So in case of Alternaria brassicae or in case of Alternaria brassicicola, so symptoms they are more or less black, and zonation it is seen in all of the cases. So, but in case of infections due to Alternaria brassicae, so in case of Alternaria brassicae, this is infection due to Alternaria brassicae. So they are smaller in size. They may be lighter in color, or sometimes in advanced stage. So this is the typical symptoms of Alternaria brassicae. These are symptoms of Alternaria brassicicola, and these are symptoms caused by Alternaria raffani. So in case of raffani, generally yellow halo is always associated. So these are Alternaria brassicae. This is Alternaria conidia of Alternaria brassicicola. This is a conidia of Alternaria raffani. You can see lot of morphological differences between the conidia of different species. यसको beak लामो छ, यसको एकदमै छोटो छ, यसको एकदम medium छ. There are other so many differences in number of septa. कतै चाहिँ transverse septa छ, कतै छैन, कतै oblique septa छ, कतै छैन. So these are symptoms on the mustard. Alternaria leaf blight of mustard, of mustard or broadleaf mustard. Rayura mustard mite is very important disease, particularly on the seed crop. So तपाईँले सजिलै चिन्न सक्नुहुन्छ. So in case of त्यही कुराहरू छ, in case of brassicae they are more dark. In case of in case of brassicicola they are more dark spots. They are more dark. In case of Alternaria brassicae, they are light brown. In case of Alternaria raffani, generally they are associated with yellow halo, so that's why they appear slightly yellowish donations. They are seen in all of the cases. Seed infections may take place. Some dark spots they may form on the seed. Pods on the seed itself. Germination is reduced excessively. So with profuse of sporulation, everything may get dark. So on cabbage, cauliflower, broccoli, it is a very it is also problematic. So generally, this causes premature leaf defoliation. So symptoms due to Alternaria brassicicola, मैले भनिसके, they are dark in color. So to remember, you can relate to Coca-Cola. So Coca-Cola, it is dark. So more or less, they are very dark color. So about conidia, their beak size is extremely reduced. So these are infections due to Alternaria brassicae. So they are also known as gray leaf spot because they are more or less grayish, and their beak size, the beak size of Alternaria brassicae, it is quite long as compared to other species, causing Alternaria leaf blight of solanaceous crop and cruciferous crop. So about disease cycle, it is also seed-borne. It can survive on the infected seeds, on infected plant debris. It has a wide host range. So sometimes on cauliflower head and cabbage head, some kind of black sporulating mass is seen. So this kind of thing, it may be it's generally due to Alternaria. So several species of Alternaria, such as Alternaria alternata, may be there, Alternaria solani may be there, and sometimes this may be also due to some saprophytic fungus such as mucor and rhizopus. So here you can see some typical spots made by Alternaria on cabbage heads. So generally temperature conditions for all very long period of time. So generally, both of you saying rhizome rot, they are seen in rainy season. So generally, the first infection that takes place on the collar region of the pseudostem. So there's some kind of water-soaked dark areas in the collar region. They proceed upward, they proceed downward to the rhizome, and they cause soft rot. You know, so you can see soft rot like thing here. So in case of fusarium, so some kind of pink, when you cut the rotted or infected rhizomes, you can see some kind of some hint of pink color due to like lycomaris meme and toxin produced cause of fusarium, so due to which there's some kind of pinkish color inside the rhizome if there is Fusarium solani. So this is one of the very important problem, and this rhizome rot, it can also occur in the storage condition. So field it may appear healthy, but when the storage conditions, they are very damp and wet, so this problem may be seen in ginger and turmeric storage as well. So apart from collar rot, so aerial on upper canopy of the plant, so some kind of yellowing, and gradually some kind of wilting is also seen. So that wilting and yellowing, that it is more pronounced in case of Fusarium solani, or sometimes there's also Fusarium wilt, that is Fusarium caused by Fusarium oxysporum forma specialis gingeri. So different kinds of Fusarium species because Fusarium oxysporum, it also can cause this problem. So in case of Fusarium, you know about light yellowing of the tips. So generally yellowing, it is initially seen on the lower leaves, and it gradually proceeds to the upper leaves, and drooping is more pronounced on the lower leaves. So and when this problem is in the field, one can easily take out the plant. So तान्दाखेरि सजिलै जुन जहाँनिर चाहिँ जुन गानो छ, rhizome छ, so it can be easily detached, so it can be easily taken out. So because this region is generally infected, collar region, so there may be some kind of water soaking, both in case of ginger as well as in case of turmeric. So as I already said, this is one of the very important disease because the inoculums, they are soil borne and can persist in the soil for a very long period of time. It may be due to spore in case of pythium. It may be due to in case calamidospores. They are also found by pythium, and they are often found by Fusarium species. So एउटा त्यो soil borne inoculum को समस्या, and generally in our case, so there is very the certification program for rhizome of turmeric and ginger, it is very poor. I know our case certifications are in case of cereal certified seed, but in case of ginger and turmeric, certification system of the propagative material, it is very poor. So generally, due to which most of the time farmers they plant diseased rhizomes, or they keep